So class, very good evening. Very warm welcome to the MBBT course. This is day one. Myself Simranjit Singh, your new trainer for the whole 10 days. Some of you might be from the previous batch of Principle of Genetics, and welcome to you also to the new group of MBBT. Um, Hi everyone, have a very good evening. Very nice evening to everyone. Um, So there was some chatting happening that what should be the timing. So we were supposed to meet at 8:15, but I think we can meet at 7:45 also because I was not sure that when I will be free from genetics class. So as soon as I finish my genetics class, I will like to join you all students here. Before I go ahead with little bit of introduction about myself, um, is my voice and the picture quality is all okay for today, because today is mainly the introductory class for you. Oh, it's 240p. Is there buffering options are happening or so? Because that was really buffering is there. Let me check my speed. So you can check your computer speed by going to fast.com. It can tell you the speed of your internet. So it's going, speed is fine. This is usually I have 40 to 42 Mbps. Uh, good. Um, yeah, I can see that there is little buffering happening. I don't know why. It's always buffering happening because of more students being joined, and that's why it shows like this circle. But from speed from behind is okay, and okay, good. So सर एकदम ऑडिबल आन दो some Punjabi ones there. So please maintain languages here. So we highly recommend let's speak in universal language, that is English, and then we continue with that. So students, first thing first. So all the links that all the things that we are sharing here, um, they will be in the form of video YouTube streaming. So you will have saved videos afterwards. So no issues for regarding little bit of any changes, or like you can have them later whenever you want to have them. So the link that I have shared, they will be saved here only, and most of the time you all students will be joining it, not from the outside. If they are joining it, we can report them so that our decor of the class will not be disturbed. And that's one thing. Yes, that's what I'm saying, Shadil, that whatever the lectures that we are doing today, so you can go through the YouTube videos again and you can have them anytime later. So that will be a no problem, no issue. To overgo, mm, so myself, Simranjit Singh, I'm a what I have done. What are my education's background before we start? So let me show you about my CV a little bit. So I did my education from Institute of Pharmacology and Toxicology from the University Medical Center, Göttingen. And then I had my PhD in molecular medicine and redox regulation of PP1 and ER stress regulation of connective tissue growth factor. When before that, I had my master's from Justus Liebig University in 2012 from the University of Gießen in Germany itself, where my topic was related to the metastasis, that is, moving of your cancer from one place to another inside your body. And before that, I had my bachelor's from Shri Udham Singh College from Punjab Technical University, BTech in Biotechnology. Um, yeah. So I know a little bit of Punjabi, Hindi, English, and also German to intermediate level. So I spent almost 7 years in Germany during my studies. So you can feel free to ask any question related to your abroad studying, or any queries or how the life is looks like in Germany, or anything related to that. So I will be very happy to answer them. And then after my studies, after my bachelor's, I also went for lecturership in Bayan Singh College in Gurdaspur, where I was teaching bioreactor design and environmental science and bioprocess engineering too. And before that, and afterwards, I went for as a research assistant in the University of Göttingen for 2 3 years, where I worked with the novel redox sensitive cysteines in PP1. Then after coming to India in 2017, I joined Mata Gujari College as assistant professor, where I was teaching principle of genetics, biotechnology, and cytogenetics. And further, online as I was worked at the moment. So I'm working due to COVID situation. I'm working as an online professor with the Edufabrica, where I'm teaching online lectures on MBBT, like to you students, biochemistry, proteomics, principle of biotechnology, and genetics. So we have covered all these subjects before for the 10 days workshop. So your students are coming in the next generations now. I have also published one paper uh, as a reduced activity of PP1 and alpha. So it seems like this one. So it was done in the Nature Scientific Reports with the reduced activity of PP1 alpha under the reduced stress condition is a consequence of glutathione mediated transient disulfide formations. So that was about me a little bit. Thank you so much. Love to Kashmir. So all the things that is happening in Kashmir, things get resolved, and we are all in peace with everyone. That's we seek for. Okay, so all the videos that I'm sharing today, so they will be shared with you. So don't worry much about them. So we will be sharing with you also about them soon. So today is an introductory class. So I will share. So I'm very glad to meet you all. Eligibility criteria is mainly to check your bachelor's grade, CGPA, your IELTS score, and if you have any experience in some specific modules of your studies. If somebody is going for material science, they will look for mathematics, IT skills, and all these skills will be checked out. So please take care of that while going for your studies. Yes, you can go for a PhD. So for that, you have to ask your professors. You have to write an email to the professors for the concerned positions, whether the things that you are doing in your masters is it eligible to continue as a PhD there. So IELTS will be mandatory if required by the, yeah, by your professor. Or for the masters, it's recommended. It is necessary, but for the PhD, it will be not. Guten Abend. Yeah, go for it, Anukriti. It's the best place to study. Also, India is not bad. I'm more patriotic to India, so I will also say that you can go for your PhD here in India itself. There is no bad things in India also. No, 3 years of bachelor's, three BSc student could also go. 4 years is better. It's always good to have more years in your education. But 3 years of studies education could also go there, so that's not a problem. And moreover, you need just good IELTS score, that is 7 band or 6.5 band in your studies, and that will be enough for you. It's because they are public university, and there is no fees you have to pay, Arunaditi. So you don't have to pay any fees there. So that's the best part. So most of the universities are public university. Don't get trapped into the agents where they will trap you to go to the private university and you have to pay higher amount. So just do your own studies. Check at that punkt day where you can check. So I will share here that. Uh, I can share one day, one class for this, how to go for studies and everything. So they are mostly depends upon entrance exam. So one question by one. Shita Yadav is doing MS Biotech like a plain degree in overall biotech a good option in Germany? Yeah, it's very good option for, but you have to learn German language while staying there, so that will open your future career. They mostly no entrance exam, but some universities like Technische University München, so they take some exams. So you have to give the exam for particular university, but most of the time it's direct admission by Uni Assist. So you want to go for Shivani Gadbole biomedical science program. Just check online. There are many websites. Bestmastergermany.com, bestmaster.com. So I can share some websites with you. One is master portal. Best university in Europe. So these are some links you can look for your studies. So I will share on this group on the, so there is a Dad.day and best universities in Europe. So there are many links actually which we can go through and look for you. So what will be the best field for topic for doing PhD zoology? But you have to look on your own. So zoology could be done in the field of insects, na? So something with the Drosophila. So I think Drosophila mangoster, the fruit fly, is a good subject to study with, and look for some particular genetic disease that you can answer it with the help of that. So look for that also. Yeah. So this course has been created actually MBBT. I will share in this course molecular biotechnology and biochemistry techniques courses. So we will deal with variety of techniques in this 10 days course, where we'll be dealing with not only techniques but some basics about the principles. What is the basic principle about it? So let's discuss soon about it. How we gonna proceed with that? So I was just So let's continue slow one by one. Yeah, there are a lot of questions. Let me go through one by one. Dad is really competitive. It depends upon if you have a good grades. If there's any scholarship from Dad, then you can go for it. Why not? But molecular medicine includes everything, basically all the medicinal diseases, medical diseases like cardiovascular disease or neurodegenerative diseases, or brain-related disease or heart-related diseases. So these could be healed with the help of molecular biology, and something related to that is known as molecular medicine. So I worked with the redox biology and cardiovascular physiology. So I was working to interact with the heart failure. How can I heal the heart attack, like if there's anyone having heart attack, so how we could recover that patient earlier state. So I was working with that. So somebody working with the brain diseases like Alzheimer, schizophrenia, or any other diseases, so they can work with that also. So that's what it's deal with. So Atre Majumdar, so sorry if I spell your name wrong, Majumdar students. So bache, so you can, there are no rules. You can go for postdoc or go for some industrial training, but you need to apply from here only in advance. That's one key thing. If you can apply in earlier days, that will be really beneficial for you. Yeah, those are the links that I share to know about the universities. IELTS is easy, बच्चे Ankush. IELTS is quite easy. Just it's a English exam that we all know. So with IELTS exam, I can share one session with you for half an hour where we were dealing with how I approached going abroad and how I did everything on my own. So don't worry about it. Yeah, papers are always good. Milan, papers are always good. If you have publications in your field of studies, you can have a better scope in the future. No, paper is not must for PhD. A master's, simple master's is enough. Good master thesis, where you have a good recommendation from your professor who have worked for your master thesis. That will be enough for you. So that will be not a big problem. So that's not a big problem. So without publishing a paper for your PhD, you can still do it. Vanishka, she's asking that we can start with the session for these queries. Yeah, that's true, students. So I will just see your questions and summarize everything in one. So keep your questions with you. I am here only for 10 days, so we can clear things with you soon. So I have seen courses of interest in both Germany and Ireland. So what their purpose in Ireland and Germany? Ireland is good place, but I would recommend Germany. But because of the future perspective, future Germany is really good in R&D. Scholarships are always there. Just go and check at the DART website that I shared you. Is ETH Zurich is a good university? We have gone through after 3 years. Do we have extra years there? What was the cost of attendance per year? Prox. ETH is good. I highly recommend that. Cost will be a lot because it's in Switzerland, and moreover expenses are high for the students. So please take care that where Germany is quite free for the students, it's a public university, they are providing you free travel also. What are the study related to requirements study masters? It's one of the best place, Germany, for your masters in neuroscience. So I will take the last few questions. What are the career options after BTech in biotechnology? Yeah, Shadil, this course will help you. This is very nice question. Um, I mean, this will be, you will have one certificate from us, whether you're taking training or not, I will come to that point. It will really help you because this is, first of all, we are doing these courses in this COVID time. So we are away from our houses. So, but you will learn all the basic techniques which could be help wherever you go for your master's or your PhD or as a laboratory technician. But for that, you have to do a very specific course that is MLT or BLT, the basic lab technician course. So for that, please consult the concerned department. But this course specifically can give an edge over the other students who are applying for any technician jobs or any research associate jobs in their level. Go for bioinformatics. Bioinformatics is huge at the moment. I will not say biochemistry doesn't have huge demand, but on the other side, because of the data science and IT department being increasing, machine learning and AI, artificial intelligence, it's in really demand. Go for bioinformatics if you have a chance. So it depends. Immunology, cancer biology, or cell biology. All these things are important for you, so you can think for your own interest. MS from Netherlands. Go for Wageningen University. If somebody is interested in agriculture, I will highly recommend Wageningen University um, for their further studies. Highly recommended. So on that note, let's stop the question answer sessions now here. Oh, that's good. That's good. So let's see. You will see by the 10th day how this course will help you. Don't worry. So let me continue with the things now. So there are two things we have to see during this course. One is assignment, and one is for the internship students review paper. Okay. Assignment is a thing in which you have to write uh, about what do you say, a summary of 2 day paper, a summary of whatever we do in a day, and write them in a summary in two-page paper. That's it. That's will be your assignment, and this is not compulsory, but I will highly recommend that if you write one, it will be really appreciable for us. I will highly recommend if you can write one, it will be highly appreciated for us. And second thing is assignment internship review paper. You have to write in a format like this one. So a review paper is something like you have to write everything in summary after reading a couple of papers, around 20 or 30 papers, as much as you can write the review. So as much as you can read it. So basically in this, on the title page, you should have your title, your name on there, date, an abstract, then an introduction, body, conclusion, and the literature cited. So all these parts have to be taken care when you are doing your writing review. So in the abstract, it should contain around 200 to 300 words. It should include the summary of review question, conclusion of your studies, and it should not cite references. Yeah, so this you have to take care. And in the introduction, you should have the topic of the study, which serve as its identification sentences. Okay, and then it should indicate the articles which you're containing, clearly outline the order in which every subtopic will be discussed to give reader background information needed to understand the section in the articles. And then in the body, you should have the subtopics of your addressing. So if you're writing about any one particular disease or any one topic that you have taken, so these things will be taken care while doing so. And further, in conclusion, you should briefly state your rationale for your review and the purpose of the article. And literature cited, use a standardized sometimes our tongues misplaced, standardized reference systems like EndNote or another reference system. So please do take care while doing so. Thank you, Ram, for sharing that link. So this will be the review format. I will share it with you. So format I have shared now. So you can write this in the group also. If you're in the group, students, please write it in the group also. So like 2 or 3 students above can write them in the group. So more than 2 or 3 students, they can write it. So, and what are the topics that you should include it while writing the review? So these are the some of the topics like gene therapy, RNA interference, genome editing, drug delivery, DNA synthesis and sequence, stem cell research, maturation of sperm cells, genetic engineering, monoclonal antibodies, cancer cell research, primary cells and transfected cell lines, cytokines and growth factors, cellular processes with growth to achieve the appropriate size, predictive models for cancer cell lines, line specific dependency. So these are some of the topics that I'm sharing with you, but it's also up to you. You can change them later part. You can have your own topics also, so that's not compulsory that you have to choose from them. So this is one of the things that I'm sharing with you now, so that you can have it, some kind of thinking that where should you go, because this is molecular biology techniques and biochemistry techniques, so these two together have to be somehow paths. And where you have to share at info@edufabrica.net, and also share on my email address. And where you can send these journals, basically you can send them, okay, not this one. So you can choose these journals if you want to publish them also. Just check them out, their policy, send to the editor, and you can publish on your own also. So I've shared all the free journals articles that could be, I don't know if they're free or not, so you have to check them by yourself. So you can contact me on the group also, or contact me via email. So I have shared my email ID so that you can look for it. You can also publish, that's what I'm saying. So I shared some journals where you can publish them also afterwards, so that you can have some benefit out of this workshop. So that's what I really want that you gain out of it after this. So for the so I will share some examples first of all. For the assignment thing, which is not compulsory, but if you write, I will be very happy. So like some student, they send me each day or they send me all together. You can send me either one day of your work to me on my email address, or you can send each after the 10 days over, you can send all them together. So what you have to write is summary, like you write, oh, my battery is getting less. So some summary like this. So students were writing in the previous classes. So I will share one example with you. This is not one of the best assignments so far, I have to say. Maybe so just have to be summary of 2-day work all together. So I will share one of the assignments with you. So this is okay. So some students worked. So I'm sharing one of the students one day. So this is done so that whenever you are going for an interview or anywhere, so you will have your own data with you so that you can read it from it. So it is done, these assignments are done mainly done for your own check, so that you can have a better check on yourself that you are going on the right path. So these files are for your own good, for your own purpose. These assignments above files are is an example of assignment. And for kind of research paper, so I will share one review paper with you. So something like this. Okay. So how a review paper looks like. So you should not be compulsory that it has to be like this. You can also write in a long format. So there's nice names. 2 students wrote it. So abstract, introduction, then parts of it. Some body part is coming up with various types. Then yeah. So conclusion is there. Then at the end, author contributions, references are there. So like this. Very nicely, neatly done. So I can share one more article with you so that you are on the good track later on. So date to submit it, so if we are starting today, 25th of October, so let's say we finish on 3rd of November in 10 days. So we can be, oh, today's Dussehra. Happy Dussehra to everyone. I totally forgot while doing so. Happy Dussehra to everyone. May all the bad things should go away from our sides, from the inside and outside, all the evils. And we are having a good and life full of joy and happiness in upcoming time. So now come to the review date final. So you will get your internship certificates only when you will submit your review. So today is like 3rd of October. So you have to submit after 5 days. So maybe on the 9th of November you have to submit everything. So 9th of November is the final date to submit review. Okay. So I hope most of your questions are clear now. So I will share my email ID here also for your students. So I have shared your Diksha review paper also. So I am giving you these 10 days also from today and also 5 days further. So you have 15 days to write your review paper. all in all. So first of all, you can be in two groups also, of 2 to 5 groups of students could write it. 2 to 5 is enough, but if you want to write voluntarily on your own, you can write it on your own also. So that's not a problem. No, only one review paper for internship students. Only one paper is enough. No, we will not finish in 3 hours. So today is an introductory day. So I will start some with introduction about MBBT, and then we start, because a lot of students write a lot of queries later on because it seems quite confusion and things are not up to the level. So that's why I take yeah, like this one, Sirikar. In internship, what's going on? What's happening? Anyone guide me properly? Anyone for group? So there are all kind of students who will get panic and who will be like fine. So that's why I took a lot of time and give my energy to make things clear for each and everyone. Yeah, Shrikar. Thing is, we will be doing an review paper internship, so which I have shared the information above, where you have to write a review paper like one of these two papers, and the format I have shared, and you can also publish it later and send it to us on this website. The format should be like this, and you have and there are topics of for internship you can choose, or you can choose your own topic also. Yeah. So just make group students, so even you can to be 6 to 7 students, I will not, it will be not bad, but thing is you should give your inputs to it, so that at the end you are all on the safe sides. Yeah, or not only the above topics, you can choose your own topics also, I will not mind that also. So there is no specific topics, you can choose your topics. If you're for training, then it's not required for to write it, so you can also skip it, but if you want, if you're interested to write to have an experience, you're most welcome. There are so many questions coming so fast. So what is a review paper? I don't know exactly what that would be, so I have explained this. Is there any specific topic? You can choose any topics, bache. How to submit it as any app for it? There is no app for it, you just have to send it by email. I mean, we are only training but not internship, then we need to do the same. No, you are not allowed to, you are not concerned with that, then you are not supposed to do it. You can skip it, that part. Purbali Mitra. Yes, you can send that also, it will be great. Dr. Shukani Bhattacharya ji. You can also send a review article of already published paper. I mean, because your training will be written that in the internship letter that you got this review article while doing an internship, molecular biology and biochemistry technique. So if you can write something related to molecular biology and biochemistry techniques, so it will be highly appreciable. No, it will be not. So you will get your own internship letter. If you're in the group, your value of your things will be not reduced. Okay. You can make group by yourself, that's fine. Okay, now, so we are done with the introductory part today with lots of questions and answers. Kshitij Yadav. Do internship students have to submit assignments compulsory? Now, assignment is not compulsory, but if you submit it, I will be highly glad. I will be very happy to have your assignments. It's good for you and for your everyone in your future. Review itself is internship. Who has paid for the internship, they have to write a review, bache. So let's start the topic. Oh man, there's so many questions coming up. Okay, it's happening. So trainees does not have to write the review, but if you can write, it will be also nice. There's no problem in it. So there's a Polemy Goswami. She said on the group that she has experience with writing review papers, so she can help if anybody needs. So be in her group if somebody doesn't have any experience, and join that group then. Thank you, Polemy, for your great concern. That's great. Okay, so let's stop talking on the WhatsApp and now concern on the things. Okay. Whew. You guys ask lots of questions. So I will be going through very basics today. It will be very funny class. I will take you to your 12th and 11th class level. So because these are very necessary before we go to the advanced level, because I want to have your basics to be clear today. Otherwise it will be of no use if your basics are not clear. So cell is like a factory. So this is an analogy that I have found that I have want to share with you all students, that cell which is inside our body, which is acting like a factory. How it is acting like a factory? So we will go through it. So in a factory, you may have discovered many different tasks being completed in the same building, such as building support shelters of workers, workers build goods, maintenance crew clean up the messes, shipping departments get ready to leave the factory, goods ready to leave the factory, and so on. And the cell also same way is working in that case, with different departments, each performing specialized tasks. And the cell wall is like a security gate, and the security gate protects the cell and helps keep it safe. And cell wall does the same thing for the plant cell. So cell wall act like a cell gate, like this one. And cell membrane is like front doors. The doors of a factory regulates what enters and leaves the buildings. A cell membrane does the same thing for plants and animal cells. So like if somebody wants to enter, the front door will be there, so they are knocking it, and only then they can enter inside of it, the cell. Whereas nucleus is like a big boss, that is CEO. The boss controls all of the activity inside the cells. Nucleus does the same thing for plants and animal cells. So And where the cytoplasm is a factory floor, where the floor of the factory has all the workers, machines, and equipments on it, and the cell cytoplasm holds all the organelles, cell parts in the plant and animal cells, like this. So all the cytoplasm is like a factory floor. Endoplasmic reticulum is like assembly line where workers do work their items, move along the conveyor belt to different parts of the plants. And ER same way work as the same way in the cells. So all the conveyor, so it's helping in your moving of your things from one place to another. And ribosomes, they are like workers on the assembly line. They are responsible for building the goods in a factory, and the ribosome assembles and they build proteins, so like this. And whereas vacuoles, they break rooms. In a room you have a storage for food items and places to get rid of the trash. Same way vacuoles holds your wastes and store water in plants and animal cells. And which cell has one large vacuole? It's a plant cell. So they are the place where you have a drink your coffee and eating and relaxing place. All the waste is being there. The lysosomes are the like janitor crews. They clean up, break down, repair all the things that is around the city, around your factory. And the lysosomes do the same things in the cell. So they're cleaners and janitors, the lysosomes. Then comes the mitochondria and chloroplast. They are like the power stations which supplies energies in the factory. So mitochondria and chloroplast in animal cells, mitochondria and chloroplast in the plant cells, they do this for the cell. So these are some of the examples. So here you can see there is a entrance by the membrane, there are doors, and then endoplasmic reticulum, it is for the entry of the things going good, and then Golgi apparatus is there. Then there's a protein and ribosome factory is there, mitochondria is there, and nucleus is there, chromosomes are there. So all are doing their own job as per the things are required. So mitochondria acting as a ATP machine, providing energy to the fuel to the machinery, and nucleus is the computerized center part where all the maps and everything is being governed, and so on, so on. So you can do it in this one page. So you're going to design a factory like that makes something. You must include the organelles from the cell like factory in the worksheet, and picture used for organelles must represent the job as well as a specific type of factory. So let's say if a ketchup factory you are making, the protein should be the bottles of the ketchup in that. So that was something related to introductory part of today of principles. Oh, this is not yours. About the introductory about factory class. So any questions so far, students? Okay. So you can write handwritten assignments also. I have no problem with it. You can send it by handwritten also. There's no problem. So that was something about introductory biology. Now a little bit about basics of molecular biology. I hope you have studied them before in details, but my reason to teach you all this that some students might have forgotten, so that you all come to the same level before we start. So that's why the first day I keep it for the very basics from the junior level, which we are learning in the 12th class or 11th class, so that you're all are concerned, you're all shared the same level of things together. So let's start. So they are eukaryotes and prokaryotes. So eukaryotes are the one which have plants, animals, and fungi, whereas the prokaryotes includes your bacteria and cyanobacteria. There is no cell wall present in the eukaryotes. In the prokaryotes, there is a cell wall presence. In the chromosome segregations, mitotic spindles, cell membranes are there in the prokaryotes. Meiosis is not present in the eukaryotes. Prokaryotes, it's absent. Ribosome size are 80s and prokaryotes have 70s. And cell organelle includes that are included in the eukaryotes are nuclear membrane, endoplasmic reticulum, Golgi apparatus, mitochondria, and chloroplast. Whereas nuclear membrane is absent, but rest of the Golgi apparatus, mitochondria, chloroplast, they are not present. Then molecular biology definition. Molecular biology is a study of a molecular underpinning of the process of replication, transcription, and translation. So whatever the things that is concerning with the DNA is replication, then further transcription of DNA to mRNA, then mRNA is producing the proteins. So all these studies and study of this molecular aspect, that is known as your molecular biology. This fields overlaps with the other biology and chemistry techniques such as genetics and biochemistry, whereas molecular biology chiefly concerns itself the understanding of the interaction between the various systems of the cells, including the interaction between the DNA, RNA, and protein biosynthesis, as well as learning how these interactions are regulated. Much of the work in molecular biology is quantitative, and recently much work has been done at the interface of the molecular biology and computer science in the bioinformatics and computational biology. Since the late 1950s and early 1960s, molecular biologists have learned to characterize, isolate, manipulate the molecular components of cells. Organisms include DNA, genetic information. RNA is a close relative of DNA, and the proteins major structural enzymatic type of molecules in the cells. So these are the main components of your molecular biology. This is our DNA, RNA, and protein. So from your DNA, with the help of transcription, RNA, mRNA is made, messenger RNA. From there, you make a protein with the help of translation. So that's the central dogma of your life. This is known as central dogma of your life. From DNA to RNA, RNA to protein. Then comes the unit of heredity. The DNA segments that carry genetic informations are called genes, and it normally stretches the DNA. So here you can see that there is a chromosome, there is a cell, then the cell there is a chromosome. Then chromosome is made up of genes, and further genes consist of DNA, which is made up of 4 chemical letters, that is A, T, C, G. So these are various gene 1 is present there, gene 2 is present there within that site of chromosome. So gene holds the information to build and maintain organism cells and pass genetic traits to offsprings. Then deoxyribonucleic acid, DNA. DNA is a nucleic acids that contain the genetic instructions used in the development and functioning of all known living organisms and some viruses. So DNA is a set of blueprints needed to construct other components of cells such as proteins, RNA molecules. 2 long strands makes the shape of a double helix. So this is the DNA nucleic acid that contains the genetic informations, and further for the proteins and RNA molecules. And this is the DNA is a mix of sorry. 2 long strand makes the shape of double helix. So this is your double helix structure, and they run in the opposite strand, therefore anti-parallel structure. Chemically, DNA consists of two long polymers of simple units called nucleotides, and with backbones made of base and sugars and phosphate groups. So you have phosphate groups on the side like there. So you have hydrogen bonds in between. So A and T, they are making double bonds, and G and C, they are making triple bonds. And this is how they are all get together. So chemically, DNA consists of two long polymers of simple units called nucleotides with the backbones made of base sugars and phosphate groups. So this is your sugar plus base, nucleosides on the left side nucleoside, and this is the base, and they together they make nucleosides. And whenever phosphate plus sugar plus base are made, they are called as nucleotides. So that's the difference between your nucleosides and nucleotides. So you have a phosphate group to your pentose sugar and nitrogenous base pairs ATCG. Then there are nitrogenous bases are of two types. There is purines and pyrimidines. Purines are fused 5 and 6 membered heterocyclic compounds, that is adenine and guanine, like this one here, who have these two 5 and 6 membered heterocyclic compounds are there. There is a carboxylic group is there, and there is a amine group is there. Amine group is present is there also. So making disulfide bonds here, so adenine and guanine. Whereas there is a cytosine and thymine, which is a 6-membered rings chains, which has carboxylic group like here and there on both sides, and also one single methyl group here present. So that's how the all the different groups are differentiated from each other. So this is cytosine, this is threonine, and this is uracil. So this is somehow misplaced up. I have to make it up. Then comes fifth pyrimidine base pairs called uracil. Usually takes the place of thymine in the RNA. Differs from the thymine by the lacking the methyl group on its rings. So they're pairing like A pair with the T and A pairs with the U in RNA side. This is in the DNA, and G pairs with the C with the triple bond. So how hydrogen bonds are fixed? So hydrogen bonds looks like this between the two base pairs, adenine and thymine. So they are making this hydrogen bonds like that, and whereas in the case of pyrimidines, guanine and cytosines, they are making triple bond like that. So, and also the one major difference between DNA and RNA is the sugars. So that we have already learned quite number of times before. The mainly reason is a deoxyribose sugar in the DNA, and in the RNA is the ribose sugar. So with the two deoxyribose sugar in DNA being replaced by the alternative pentose sugar, ribose in the RNA. So here you have deoxyribose sugar, the second 2-deoxyribose is removed, oxygen is removed, and in the ribose sugar in the RNA, this oxygen is present in the second carbon atom. And this is the size matter. So DNA chain is around 22 to 26 angstrom wide. They are that much of wide, and then like 2.2 to 2.6 nanometers diameter, and one nucleotide unit is 3.3 angstrom. So the distance between each base pair is 3.3 angstrom long. And there is a major groove, minor groove. These are base pairs, and on the side is a sugar phosphate backbone group. So ribonucleic acid RNA is biologically important type of molecule that consists of RNAs. So before we go ahead to the RNA part, let me go through the previous questions so that we are on the same level, each one of us. Do internship students have to submit assignments compulsory? Do internship students have to submit assignments? No, assignment is not compulsory, but if you can do it, then that's good. Raksha Jain, no, both registrations are not same. If you are trainee, you are not part of the internship. If you want to still be part of internship, you can register there to do so. You are doing internship here with us only. which topic we have to make assignments, how to send it. So assignments, bache Shadil, is same, like you have to write a summary of each day and of 2 pages. I have shared that in the WhatsApp group, just go through it. Otherwise ask in the group, somebody could help you then, for sure. Where you have to submit the assignment, you have to submit the assignment on my email ID, simranrehals@simranrehals99@gmail.com. How is this training and project associated with IIT? So we have a with IIT Kharagpur, we have a team, like proper what do you say, bond registered, that these internships will be signed by the IIT Kharagpur, signed with you. So we are in collaboration with them. So whatever the certificates and yeah, certificates that you will get, you will be signed with the help of IIT Kharagpur. Cell and molecular biology, when we are working in the lab, it mean different in the techniques or methods. But cell biology is something that we are more concerned with, the cell organelles. So about how the trafficking of the cells is happening, how the Golgi body is working, how endoplasmic reticulum is helping in the trafficking of your mRNA. So we are more concerned with that part, the cell organelles. Whereas molecular biology chiefly deals with your how we can extract these parts out of the DNA of your cells and look for their functions. We are mainly then working at their interactions and how DNA is working out, how RNA is working out. So we are mainly how protein is working out. So we're dealing with DNA, RNA, and protein, and how they're interacting within the cells. So that's what the molecular biology is dealing with. Yeah, I can understand. It happens sometimes. All these terms are coming together. So, okay. So let's go for the next part then, if you have all clear till now. Now RNA. So RNA is a biological important type of molecule that consists of long chain of nucleotide units. Each nucleotide consists of nitrogenous base, ribose sugar and phosphate. So it's the same like DNA. You have a ribose sugar in this case and phosphate group and nitrogenous group. So this is one example of that. So they are not double chain. So they're single. Then there's a double. Then there's a single. Then there's again double chained. So like this, this single and double chained pattern of your DNA is made. So double-stranded RNA is RNA with 2 complementary strands similar to the DNA found in all cells. Double-stranded RNA forms the genetic material of some viruses also. So like this HIV virus. So what are the types of RNA in total? So they are messenger RNA, ribosomal RNA, and transfer RNA. Abbreviations are mRNA, rRNA, and tRNA. So codes for proteins. So the codes for proteins, messenger RNA, ribosomal for translation, and transfer RNA also for translations, and they are present in all organisms. Then comes in the post-translational modifications. They are small nuclear RNA, that is known as sRNA, and their function is splicing and other functions, and they are present in the eukaryotes and archaea bacteria. Then there is yRNA. They are help in the RNA processing and DNA replication. They are present in all animals. And there is a telomerase RNA also. They are present for the telomerase synthesis and most eukaryotes. And regulatory RNA, antisense RNA, aRNA, transcriptional attenuations, mRNA degradations, mRNA stabilizations, translations block. So all comes together, and they are present in all organisms, antisense RNA. So messenger RNA carries information about protein sequence to the ribosomes in the protein synthesis factories in the cell, and it is coded so that every 3 nucleotides responds to 1 amino acids. So like this, here you have your DNA being unspliced, so they are open and winded out, and you have coding strand, you have messenger RNA. And then you have non-coding strands here, which is coming out of the 3 codon structures like this. So in eukaryotic cells, one precursor mRNA has been transcribed from DNA and it is processed to make mature mRNA. And in this processing, it removes introns, non-coding sections of the pre-mRNA. So all the non-coding sections, that is introns, they are removed while doing so. Then this mRNA is exported to the cytoplasm from the nucleus, where it is bound to the ribosomes and translated into its corresponding protein with the help of transfer RNA. Whereas in the prokaryotic cells which do not have nucleus, cytoplasm compartments, so mRNA can bind to ribosome while it is being transcribed from the DNA. So that's the major difference between your eukaryote and prokaryote, how the transcription factor is being changed. Then comes the transfer RNA. Transfer RNA is a small RNA chain of about 80 nucleotides that transfer specific amino acids to growing polypeptide chains at the ribosomal protein of synthesis during the translation. So it has sites for amino acid attachments and anticodon regions for the codon recognitions that signs to the specific sequence of messenger RNA through the hydrogen bonds actually. So here you can see the structures. You have intermolecular base pairings. When they are double paired, their intermolecular base pairing is happening. So you have by ester bond, you have amino acids group attached to the top. On the top of your tRNA, and the bottom is your mRNA, which is coming down and getting attached to your codons. And this is how your transcription, translation and translation will work out. So then ribosomal RNA is the catalytic component of the ribosomes, where your transfer RNA will attach actually, and there are 4 types: 18S, 5.82S, 28S, 5S, and they are synthesized in the nucleolus. So in the cytoplasm, ribosomal RNA and protein combine to form nucleoprotein called ribosomes. The ribosome binds mRNA and carries out protein synthesis. Several ribosomes may attach to single mRNA at any time. So ribosomal RNA is extremely abundant and makes up 80% of the 10 mg per ml RNA found in typical eukaryotic cytoplasm. So difference between RNA and DNA is RNA nucleotides contains ribosugar, it has deoxyribosugars, it has base uracil, it has base thymine, it is the presence of hydroxyl group at the second position of the ribosugar, it lacks hydroxyl group at the second position of the ribosugar. RNA is usually single stranded and here is a double stranded. That's the major difference between your DNA and RNA. So before I go ahead, let's go for small videos. So when videos are playing, I can't speak. So when the videos will stop, I will start speaking.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order, but small differences in the sequence of the bases in our genes make each person unique.
So that was something about how DNA and genes makes a difference between each of us. So how same genes, different organisms could play a role in our life. So let's go through that.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
So that's how the difference of same genes in different organisms, you can use it in a very wide manner of applications. So let's build a DNA molecule. So T is there. What should be the next part of it? It should be A. And G, if it is G, then it should be C. And G, then C. C, then G. T, then A. C, then it is G. T, then it's A. So like this, our DNA molecule being constructed. So I don't know for how long I have to do this. Oops, C is G. A is T. A is T. T is A. G is C. T is A. So at this rate, I need to work day and night for 50 years to transcribe the DNA in one human cell. So this will take around 50 years to do so. Clear? So yes, Anuriti, all the PCR centrifugations, they all deal with the molecular biology part. Mostly 15% is fine, but I can accept 25% also. Even the research papers, they accept till 15% of your plagiarism. So students, please take care of the plagiarism also. Plagiarism is something like you copy from one paper and then you paste it just like that. So just take that. You have to write in your own words. Everything has to be fresh and you have to take care of while writing. So Yes, they are in collaboration with IIT Kharagpur, so you will get the certificates with their presence. What is the function of mRNA and siRNA? So, बच्चे siRNA is known as a small interfering or short interfering RNA, whereas mRNA known as a microRNAs. So, siRNA is not conserved throughout the species, while mRNA is highly conserved and related to the organisms. So, siRNAs, they are cutting at the specific part of your work that you are working on, and so just that part. So Atriya Majumdar, sorry if I spell your name right. Please ask in the group. So some of the students, they are writing in the group, in the WhatsApp group. Check that. So students are already answering that. So that's how we build the DNA. uh, so this was 3 parts of it so far. So now next, the last thing that I want to show you is second last maybe.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
So now I will explain you about how to transcribe and translate a gene. So whatever I have taught you about different RNAs and different ribosomal RNAs and tRNAs, so how they are working together.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life. Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So let's give a transcription a try. Remember, A in DNA pairs with the U in RNA.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So GGA, what it should be? So what should be the anticodon for GGA? So it will be CCU, glycine. Then GGU, CCA. Then CGG, CGG would be GCC. So like this we continue.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, DNA helicase, will now slide between 2 complementary strands of DNA, temporarily separating them so that they can be copied. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So that was about transcription. So that was about transcription and translation. So we are done till here. So we had quite nice videos about so far what we have studied, and this is how we will go. We will go through some literature and some nice videos, some virtual labs also, so that we can have a good overview of everything. And students, if some of you have not subscribed to my channel, please subscribe to the channel also. This will really help me to grow my channel and to teach you more students, to bring more things for you, to bring more achievements and bring better things for you. So please subscribe to the channel, and yeah, and enjoy some videos like this. I will try to make some animated videos soon. Just waiting for right time and right place to do so. So let's continue. So proteins are the polypeptides. They are made up of amino acids arranged in a linear chain and folded into globular form. And the sequence of amino acids in a protein uh, sorry. In a protein is defined by the sequence of a gene which is encoded by a genetic code. So genetic code specifies 20 amino acids. So this is how your proteins in present in the fish, meat, egg, chickpeas, peanuts, red kidney beans, all are present in the in this case. So all the amino acids, polypeptide chains of amino acids coming to form this polypeptide chains and coming together. So basic players in molecular biology that are playing are DNA, RNA, and protein. So this is known as your central dogma of life. This is known as your central dogma of your life. So that is DNA is producing replication, could also go for replication. And then transcription will happen to do the RNA production, then protein production. This is whole known as your central dogma. Then DNA replication is something based on the biological inheritance base, a fundamentally process occurring in all living organisms to copy their DNA. In the process of replication, each strand of original double-stranded molecules serve as a template for reproduction of the complementary stranded. So two identical DNA molecules are produced from a single stranded DNA molecules. So here you can see the DNA polymerase, polymerase alpha, DNA ligase is there, RNA primer, DNA primer. So first of all, what will happen? The topoisomerase, this is the enzyme which will unwind your DNA, and then the helicase is the one which will break apart your DNA molecule. So you have two strands, lagging strands and leading strands. In the leading strands, everything will go smooth with the help of DNA polymerase. It will start producing the opposite new strand, and whereas it will start producing like that. But on the other case, DNA polymerase will also work, but some kind of Okazaki fragments will be produced, which will be separated from each other, and that will be filled by the DNA ligase and also help of RNA primer and DNA primase. So they will come together and play a role in that. So in a cell, DNA replication begins at specific location in the genomes called origins. Unwinding of DNA at the GNA at the origin and synthesis of new strands forms a replication work. So in addition to the DNA polymerase, enzymes that synthesize the new DNA by adding nucleotides match to the template strands, a number of proteins are associated with the fork assisted in the initiation and continuation of DNA synthesis. So the cellular proofreading that ensure near perfect fidelity for the DNA replication. So now comes the next transcription. Transcription is a process that we have discussed now, production of your equivalent RNA copy of sequence of DNA. So transcription is the first steps leading to the gene expression. Transcription, reverse transcription, like this. And during transcription, a DNA sequence is read by RNA polymerase, which produces a complementary anti-parallel strands. And transcription results in an RNA complement that includes uracil and instead of thymines here in this case. So I will not go through in details about this transcription. We have discussed already how transcription is done with the help of messenger RNA. So here is one diagram. DNA is reading from 3 to 5 to 5 prime end during the transcription. and complementary RNA is created from the 5' to 3' direction. So you have your RNA polymerase and your DNA is unwinded, and then there is a termination signals at the end and self-complementary sequence. So from your DNA, your mRNA is being produced in this way. And also you have introns in DNA. So these introns will be removed, RNA splicing will be done, and you have at the end exons 1, 2, 3 which have exact DNA information instead of your RNA information. So those all will be removed at the end of the case. And reverse transcription, reverse transcribed viruses replicate the genomes by reverse transcribing the DNA copies from the RNA, and these DNA copies are then transcribed to new RNA, and retrotransposons will also spread by copying DNA from one another. So reverse transcriptase, which transform your DNA to back to the RNA to back to the DNA copy. So this is general, normally this happens, but this is special case if RNA is producing specially to DNA or RNA is self-replicating itself. Then translation, where protein biosynthesis is happening. In the translation, mRNA produced by transcription is decoded by the ribosome to produce a specific amino acid chain that will later fold into an active protein. Translation occurs in the cell cytoplasm, where the large and small subunits of the ribosomes are located and bind to the messenger RNA. So here, like we have discussed it already, how you first transfer RNA is there. So like valine has been attached, now it is attaching to the alanine part. So from there alanine is growing up with this protein chain of amino acids, and here we have codons for those amino acids, and we have ribosomes being attached there, and this is your messenger RNA. Then comes your glycine being attached there. With the glycine being attached, direction of ribosome advance is going on, then you have a codon afterwards. So in this ribosome facilities decoding by inducing the binding of transfer RNA with complementary anticodons sequence to mRNA. And these are the genetic codes that you have been studying quite so far. You write UCAG on the left side, UCAG on the upper top, and you write 4 letters here, UCAG. Then you take first letter from left, U, from top second U, then third from U, from here, UUU. So UUU, then UUC, then UUA, then UUG. So all these comes together. And work like this. So they are start codons like methionine. Start codon is AUG, this one. Your start codons are methionine, and your stop codons are UAA, UAG, UGA. The rest are like this. So remember them as together in a group, or you can remember like this. So what is genome? Genome is entirety of organism heredity. It includes everything, all the information of one person, like in CPU of computer includes all the information, central processing unit. Same way genome has entire organism hereditary information. So it is encoded either in DNA or many types of viruses in RNA, and the genomes includes both genes and non-coding sequence of the DNA in this case. So comparative genome sizes of organisms. So Homo sapiens, so they have around 3.2 billions, mouse has 2.6, fruit fly have 137. So like this it keep going on, and these are the various chromosome numbers of them. So just comparative of genomic data of each organisms. So you can see such a wide differences from top to bottom, how from bacteria, that is E. coli, to your human sapiens. So bacteria has 4.6 millions of size base pairs of your genome size. So why to study genome analysis? So genome analysis is done to the production of genes in uncharacterized genomic sequence to obtain the complete sequence as many as genomes possible for genetic modifications. So genetic to develop new varieties at faster rate like BT cotton and BT brinjal. So some tools, some introductory tools about used in molecular biology. Gel electrophoresis that we will use, the basic principle. So I will give you just little bit about tips about them, but we will go in details in our lectures soon. So gel electrophoresis, the basic principle is that DNA and RNA proteins can all be separated by means of an electric field. In agarose gel electrophoresis, so you have DNA of different sizes, and you run over the gel with the negative and positive end. Your DNA is negatively charged because of the phosphate group, and then DNA bands are exposed, and you have yeah, you can check the size according to their length. So the shorter will move further, and middle ones will be in the center, and the longer ones will be at the top. So that's how you can differentiate between your size of various DNA or RNA or mRNA or tRNA or ribosomes, anything that you're looking for. You extract them and you run over the gel, and you can check their sizes by checking in these groups. Then comes the polymerase chain reaction. The polymerase chain reaction is an extremely versatile technique for copying DNA. PCR allows a single DNA sequence to be copied or altered in the predetermined ways. PCR has many variations like reverse transcription, PCR for amplification of RNA, real-time PCRs which allow for quantitative measurement of DNA or RNA molecules. So you have PCR analysis, there is a wanted gene and template gene. The process follows the principle of DNA replications. So this is first cycle, template DNA, 2 by 2 copies. So this is a 2 by 3 copies, and then 16 copies, then 32 copies. So you have exponential amplifications of your DNA, of your PCR analysis. So primer we will discuss. So their applications, their study of patterns of gene expressions. Tasks for the DNA sequence can be assisted by PCRs. PCRs have numerous applications to the traditional process of DNA cloning. Existing application of PCR is phylogenetic analysis of DNA from ancient things. So it has numerous PCR has numerous applications. Then for the macromolecules blotting, probing, like for DNA and RNA, if they are big molecules of that, how you can probe them? You can do it with the southern blotting. So you have genomic DNA, you restrict with the endonuclease, and then you run on the electrophoresis gel. Then with the help of blotting paper, you transfer them into the nylon membrane, and after the nylon membrane, you probe them with the radioactive probes like 32P phosphorus, which is specific to one band, and then you can expose them to the X-ray film. This was that's what I explained the southern blotting just now to you. And southern blotting is done for the DNA, and northern blotting is done for the RNA. And western blotting is done for the proteins. And their molecular markers, they are based on naturally. So students, don't get confused that I'm just separating them like this. We will go in details in one of your class is based on this. Oh, I didn't share you the objectives. Sorry. One of your lectures is based on these things. So I will, blotting techniques is day 7, so we will go in details on that day, and there is separation of electrophoresis also on one day. So that's why I am just teaching you the basic part so that you can have a little bit of awareness that what is the importance of all these parts. Yeah, so I will share this part also with you. Uh, yeah, objectives. So I also shared today's files with you just now. Also the links, maybe later I can share that part. Let me just first finish with today. So don't worry about that. So molecular markers are the one which are based on the naturally occurring polymorphism in DNA sequences. They are the sequence of DNA which have been traced to specific location on the chromosome and associated with the particular traits. And it can be described as a variation that can be observed. So these are the various genetic markers. We will also deal with each of them in our lectures. RFLP, AFLP, that is restriction fragment length polymorphism, AFLP, that is amplified fragment length polymorphism, then RAPD is a random amplification of polymorphic DNA, VNTR is the variable number of tandem repeats, microsatellite, SSRs, SNPs, STRs, SFPs, DRTs, RAD markers. So these all the various genetic markers. And the best markers are the one which are polymorphic, codominant, inherited, easy and cheap to detect, reproducible, and distributed throughout the genome. So they are could be of several applications like genetic diagnosis, study of genome, parental testing, phylogenetic analysis, selection of livestock, transformations, germplasm characterization. So let's okay. RFLP deal with the restriction fragment length polymorphism. It involves the fragmentation sample of DNA by restriction enzymes which can recognize and cut DNA wherever specific sort sequence occurs. Advantages are they are quite variant and codominant, and disadvantages they require large amount of DNA. AFLPs are the one which analyze the amplified restriction fragments, and it can be used for comparisons between close related species. Advantages are they are fast, relatively inexpensive, highly variable. RAPDs, they are the random amplification of polymorphic DNA. It is a type of PCR, but a segment of DNA that are amplified of random things. Then microsatellites are the one which are SSRs. They are used for repeating sequences of 1 to 6 base pairs of DNA. SNPs are single nucleotide polymorphism, is a DNA occurring when a single nucleotide in the genome differs between the members of species, paired chromosome individual. STRs are the short random repeats in a DNA, occurs when pattern of 2 or more nucleotides are repeated with directly adjacent to each other. So like CATG, which are being repeated again and again. They are used in the forensic cases or the genetic fingerprinting of individuals. So now last part is principle of DNA isolation and purifications, that we can have various sources include blood, buccal cells, culture cells, bacterial cells, biopsies. So mainly you have to isolate your DNA, then you would go for lysis, protein degradation, then binding is done, then washing is done, centrifuge, then elution is done, then pure genomic DNA is out of it. So that's the basic steps of your getting your DNA. So in first case, in your cell disruption, this is commonly done by grinding or sonicating the sample and removing the membrane lipids, adding a detergent afterwards. Then isolation of DNA is done adding a protease, and then afterwards precipitating it using the ice cold ethanol or isopropanol is used. Since DNA is insoluble in these alcohols, so it will be aggregated together, giving a pellet upon centrifugation. So these steps also removes alcohol soluble salts afterwards. So basic rules you have to maintain. In the case of blood, if you're using lyse the cells with the help of gentle detergents like Triton X100. Then wash your cells and inhibits the restriction enzymes and attack polymerase. And work on ice to slow down enzymatic processes. Wear gloves to protect your samples from you. And autoclave all solutions stored in fridge. Keep all pellets and supernatant until you have DNA you want. So to get your DNA, so you lyse your cells in the presence of NaCl so that DNA is stabilized and remains as double helix. You add EDTA, So to chelate magnesiums, which is a cofactor of DNAs, which chew up the DNA rapidly. Then you add anionic detergent SDS, so which helps to dissolve membranes and binds to the positive charge of chromosomal proteins. Then you add protease to that to digest the proteins, incubate the solution at elevated temperature. To getting rid of the proteins, you add organic solvent solutions, usually equal volume of your phenol and chloroform to it. And then for precipitation, you add ice cold ethanol, 95% ethanol with minus 20 degrees Celsius overnight. You centrifuge it and then wash the DNA palette, remove excess salts in 70 degrees ethanol. Then you store it in the 4 degrees Celsius, frozen at minus 20 degrees Celsius for the long term. You can quantify your DNA with the help of this formula in the spectrophotometer. Like you can have, if you have with the 260 optical density double-stranded RNA, you get 50 microgram per ml, then 33 microgram per ml, and 40 microgram per ml. You can get it. Then for the DNA purity, you can check if you have 1.6, less than 1.6, then protein contamination is there. If it is more than 2.0, then chloroform or phenol contaminated is there. Then purify your sample again. And this is how you sample of for DNA extraction: lysis of cells at elevated temperature, removal of cellular proteins, precipitation of nucleic acids, and quantification and purity measurement of DNA is done. So thank you very much, students. That's it for today. So tomorrow we will do a virtual lab. I think it's you can't be. So I will share the links of that I was teaching you today. So maybe you can go through these links later. So questions, students. When will be there attachments of DNA and RNA? I didn't get you. So when Bt cotton is genetically modified plant but it's help kill pests due to the cry gene distorting the pest digestive tract, why is that farmers are still afraid to use it in India? But it's distort the chemical that was a major discussion in Germany also. So they were creating a lot of GMOs. So but students, but the local people doesn't want to eat GMOs, and they were killing them, and it was a big havoc in there. So it's mainly the people doesn't want it because they think nature has, we should not play with the nature. As what we eating organic, we should eat organic food, not with the other part of it. So that's how they maintain it in that way. But Western blotting is done to know the protein level, to how much protein is there, to check the protein expression in your tissue level. Southern blotting is done to check the DNA level, like how much DNA is present in your things. So okay class, that's it for today. I will take you tomorrow with the same thing of DNA extraction with a virtual lab, and we will see us tomorrow. So tomorrow we will see us at 8:45, 7:45. So timings will be 7:45, and we start at 8 o'clock. So we work from 8 to 10 or 9:30 or so as per the time suggest us, and then we continue like this. So we starts at 7:45. Formation of ribosomal RNA anywhere. No, bache, this is a technique based subject. So we will mainly learn the technique. So today was mainly to have a overview of everything of molecular biology. so that you're all on the same panel. Oh, student spelling got wrong. Anyhow. Okay, students, it was pleasure seeing you. Wish you all good night. Happy Dussehra again. And it's a festive season started already. So till Diwali, every day will be festive season like Christmas we have in Germany, we used to have. So this is a span of time where we enjoy each time. So I will say good night, goodbye, and see you tomorrow then. Same time, same place, same level. Keep it high spirit, and keep in the positive vibes, and generate positive vibes, remain in positive vibes, and spread happiness as much as you can. Yeah, take care. Bye bye. Enjoy your evening. Thank you so much. It was pleasure meeting you all. Good night everyone. Good night. Adios. Gracias. Buenos Juvenos. Something like that. Good night. I will end the stream now. And please subscribe to my channel. It will really help me to go longer in my life, to start keep up my YouTube channel. Thank you very much. Thank you very much. Shall I stream it off?